we have a bit of a change in our schedule, and Tara Clark hasn't been able to join us, so we'll be jumping down to do commercial mycorrhizal inoculants improve raspberry growth. And at this time, I'd like to go ahead and introduce Quan Liu from WSU Experiment Station.
Hello everyone. It's okay to start? Okay. Hello everyone. Welcome to Small Fruit Conference. And my name is Qianwen Lu, and I'm a PhD student in Northwestern Washington Research and Extension Center, Washington State University. And I was tutored by Dr. Divider, Dr. Feng, and Dr. Tao. And today I'm going to talk about do commercial mycorrhizae inoculants improve raspberry growth. So raspberry is a really important crop in Washington State. And I think most raspberry growers are much familiar than me with the importance of red raspberry here. Like Washington State that process the red raspberry production. But I'm not going to talk about today. I just wanted to state two facts here. First one, if we apply synthetic fertilizer in long run, they will change soil pH and shift the soil microbial composition. And soil fumigation, which helps to control the soil-borne disease. But the toxic chemical will kill the beneficial soil biotest. So both of these will inadvertently reduce beneficial soil biota. Another fact is the buildup of soil-borne pathogens like root rot and root lesion nematode, they will reduce raspberry health. So the two facts here will reduce raspberry productivity. So what can we do to avoid this loss? After searching online, we find that a vascular mycorrhizae fungi could be a potential solution. So I wanted to introduce a little bit about what is a vascular mycorrhizae fungi here to get you started. So mycorrhizae, you know from the name mycorrhizae, they're really tiny, small things in the soil, and you can see them with your naked eyes. And they form symbiosis with host plants, especially a vascular mycorrhizae fungi. They form symbiosis with more than 80% of the terrestrial plants through the way that they provide nutrients to the host plants in exchange of photosynthetic products like carbon, because they cannot synthesize carbon by themselves. And their major function is to increase plant uptake of nutrients. So the graph on the left hand show you that the pathway, how they increase the nutrient to the host plants. So there's a spots like outside the root area, they will generate a hyphae first, and the hyphae will extend into the root hair and keep proceeding, penetrate the root epidermis, and then will growing in the root cortex cell. And you can see this phyla branch, this pair of hyphae are vasculars, and they are the dominant characteristic of a vascular mycorrhizae fungi. Okay, this is some results from the search online, and we can see that lots of newsletters and research articles, they documented multiple benefits of mycorrhizae, include that they state mycorrhizae fungi will help increase nitrogen acquisition from soil, and also increase the antioxidant profiles and activities in different plants and crops. So this is one of the promising results from the listed articles. So in this graph, the y-axis is plant biomass, and the x-axis is different treatment. We only focus on the control, which is shown in the brown bar in the graph, and AM fungi treatment, which is shown in the yellow bar in the graph. Here, this is a control, and this is AM fungi. And we can see that the yellow bar is significantly higher than the brown bar. And it states that the plants colonized with AM fungi are associated with significantly higher biomass than control plants. So as you have no multiple benefits of AM fungi, and now someone wanted to promote this product to you, don't just take money out of your pocket. Please wait and see if you can get some information from my research work. So as you have no multiple benefits of mycorrhizae fungi, and this is one of the factors in my research work. Another factor I wanted to test in my research is organic derived fertilizer source. Because the research regarding with the effect of organic derived source on red raspberry growth and development is really limited. And we are lucky because we have a couple of big scale dairy farm nearby, like Adeline farm. I assume they can like provide high quality organic derived manure to red raspberry with lower transportation costs. And both of the benefits from mycorrhizae fungi and organic derived fertilizer source has been characterized on the plants like strawberry, petrol, Roman lettuce, and eggplants. But the potential benefits of these two factors have not been characterized on red raspberry. So this is my research niche. So my objective in the research is to evaluate combined effects of different fertilizer source and the mycorrhizae fungal inoculants. And I used a greenhouse experiment to evaluate this combination effects. So this is a schematic figure of my greenhouse experiment. And this greenhouse experiment was conducted in our center, Northwestern Washington Research and Extension Center, from June 2019. And in the greenhouse, the light was supplemented and the temperature and humidity was recorded. And different commercial mycorrhizae treatment combined with different fertilizer treatment were applied to mycorrhizae growing deep pot through a sandwich method. I will show you what is sandwich method later, but now I will just show you, here is the deep pot. This narrow, long pot is deep pot, and they have several holes here at the bottom, and they're used for drain water and avoid to be saturating. So here I will show you what is the sandwich method. This is a deep pot, and we will fill up the deep pot from bottom to top, just like a sandwich. This is like through the way I set up my experiment in greenhouse. So the bottom soil is mixed soil medium, which is meadow, sterilized field soil, sand, turf-based MVP at a 1:1:1 ratio. And I wanted to point out here that the sterilized field soil is from linden. And the middle part, like the veggie or meat part in the sandwich, is mycorrhizae inoculation treatments. And I use 5 different treatments here. And the first 3 of them are commercial mycorrhizae treatments. As you can see, I will show you here. This is a bioorganics. And this is Indo. And third one is mycos. And the first treatment, raspberry field community, which is also the same soil we get from linden, red raspberry field. And the 5th is non-inoculated control. Here, the top bread is also the mixed soil medium. And the topping part of the sandwich, we apply different fertilizer treatments. And I use 3 of them. The first one is organic fertilizer, which I use Weiser. And the second one is synthetic fertilizer. I use the urea plus with phosphate and potash. And the third one is no fertilizer control. I wanted to indicate here that in the following presentation, I may use Weiss organic to represent organic fertilizer and urea to represent the synthetic fertilizer separately. And I also wanted to point out here that both the organic fertilizer and synthetic fertilizer have the consistent NPK. So after we set up the deep pot, the red raspberry tissue culture, maker red raspberry, was growing in the deep pot. And the roots will stretch into the mycorrhizal inoculation treatment, this middle part, they will get full contact with different mycorrhizal inoculation to form inoculants. And this is my experiment design. I use randomized complete block design, and I have 8 replications, which are 8 blocks. And in each block, there are 5 different mycorrhizae treatments, multiple with 3 different fertilizer treatments, just as I just mentioned and show you in the schematic of the sandwich method. So total is 50 different treatment combinations. And the graph on the right hand, you can show this is a whole block in my greenhouse, and there are 15 plants, and 5 of them are in 3 different racks, and they represent 50 different treatment combinations. And you can also see here it's like small labels. I label with different treatment combinations, so I can easily discern them in the greenhouse. Data I collected including the following. First, plant growth. I collect the plant height, chlorophyll content, shoot and root biomass. Plant and soil nutrients include macro and micro nutrient concentrations of plant shoots. And the third is mycorrhizae fungal colonization rate. So this picture shows I was collecting the plant height data, and this I was weighing the root biomass in our lab. And this is shows I was making the slides for observing and calculating the mycorrhizal fungal colonization rate. Okay, let's move on to the results part. Because there's still some like data being processing, so I only show you the data I have. So this shows you how the raspberry grow after as the time passed by, and you can see they are growing from really tiny raspberry in June and to really bigger and higher red raspberry in October, and some of them are even taller than 45 inches. Before I present my colonization rate data, I want to show you the procedures to observe and calculating fungal inoculation. So after the roots was activated from the deep part, I will cleaning them with potassium hydroxide first, and then staining them with trypan blue. So they will like change into the blue color. And then I make the root slide, and then I will observe and counting the mycorrhizae colonization rate on the microscope. And here is 2 pictures show you what a vasculars, hyphae, and vesicles look like on the microscope. So here is the vasculars, and they are like a flower growing on the tree trunk shaped hyphae. And here are hyphae. And this is a vesicles, and they like a bloom. And here, I'm sorry, so here, they have a very small tail, and this is a hyphae channel connected to the vesicles. And here you can see this is a vascular mycorrhizae, hyphae. They are quite straightforward, not twisted. Okay, and this is my mycorrhizae colonization rate data. So the treatment that does not form any mycorrhizae, like their colonization rate is 0. I have already tailored them from the table. So all the data left here, they have colonization rate. And this table has 2 columns. The first column is different treatment combinations. And I nested different mycorrhizae treatment within the fertilizer treatment. And the second column is colonization rate, and they denoted by different percentage. Okay, let's look on the first batch of data. So within the Wise Organic, which is organic fertilizer treatment, we can find that the bioorganics and the mycos, these 2 mycorrhizae treatments are higher than Indo and the raspberry field community mycorrhizae treatments. And also, let's move on to urea. And we can see within the urea treatment, the bioorganics and the mycos are also higher than Indo. This is a shoot biomass data, and there are two graphs in this slide, but they share the same y-axis. As you can see, the y-axis is shoot biomass here, labeled with grams, and the two different axes. So the left graph, they have the x-axis as mycorrhizae treatment, and 5 different mycorrhizae treatments are listed here with different colors. And in the right graph, their x-axis is fertilizer treatment. So 3 different fertilizer treatments are listed here. Okay, let's see this graph first. So in the left graph, we can't see any difference between the mycorrhizae treatments because they look quite similar. But in the right graph, we find that the shoot biomass was significantly different across different fertilizer treatments, especially the urea are taller than white organic and control. This is a root biomass data. So they display the same way as shoot biomass, as they share the same y-axis, which is labeled with root biomass. And the x-axis is also different in 2 graphs. In the left is also mycorrhizae treatment, and in the right is also fertilizer treatment. And we can see that the root biomass of different mycorrhizae treatment does not show any difference because they also look really similar. But bioorganics seems like to have numerically higher root biomass than other 4 mycorrhizae treatments. And the fertilizer treatment, we can see that the urea and the white organic are significantly higher than control in root biomass. But there is no difference between the white organic and the urea because they have the same like letters here. Okay, these are 3 take-home conclusions. First, plants form the more mycorrhizae with mycos and bioorganics than Indo. It's just a conclude from the table I show you first about the mycorrhizae rate data. And second, growth measurements were more impacted by fertilizer treatment than mycorrhizal inoculants. As I just showed you the root biomass and the shoot biomass data, because there is no difference among different mycorrhizae treatments, but there is a difference among different fertilizer treatments. The third one is further research are needed to justify the role of mycorrhizae on raspberry growth and development, because so far we don't find any like significant difference across mycorrhizae treatments except for the colonization rate. This is an acknowledgement, and thank you so much for the financial support from Washington Red Raspberry Commission and the Washington State Department of Agriculture. And thank you so much for my advisors, my collaborators, and my lab mates to help me make more progress about my research work. And my collaborator, Erica, she will present tomorrow and share more secrets about a vascular mycorrhizae fungi with you. And thank you so much for all of you to coming here and listening to my presentation. And I'm happy to take any questions. Also, if you have some other questions, you are feel free to email me.
Hi. I'm a little confused on this. Excuse my ignorance, but is this mycorrhizae, whatever you call it?
You mean a vascular mycorrhizae fungi?
Yeah. Is it a micronutrient like humic acid or whatever?
Oh, this is just a kind of fungi. It's just a soil organism, because the soil has bacteria, fungi in them, and a vascular mycorrhizae fungi is just a kind of fungi. I mean, actually, they are not seldom to say they are ubiquitous in the earth because as I just mentioned, that more than 80% of the terrestrial plants form the have the symbiosis with a vascular mycorrhizae fungi. I remember there's like blueberry, so specific species of blueberries, they don't have like the root hair, but they can form a vascular like symbiosis with a vascular mycorrhizae fungi. So they will like uptake the nutrients through the vascular mycorrhizae fungi, hyphae, vasculars or vascles instead of the root hair because they don't have root hair.
Now you're mainly thinking about putting this whatever they call it. Excuse me.
AMF.
Okay. As a root dip or defeed or is it a certain mixture you put into the fertilizer? I'm trying to figure out what the substance is.
Oh, so you are asking like how I apply them in my experiment, right? Like how I apply them in my experiment, right?
That's right. Like either dipping the roots or is it treatment of soil or is it a spray on?
Okay, I will show you because like this because I probably to explain this during the presentation, but due to the time limit, I just tailored this slide down from my whole slide. So I just mentioned 5 different mycorrhizae treatment I use, right? And the first 3 of them of commercial mycorrhizae. So for the first one, it should be bioorganics, but it's just write the micronized into mycorrhizae inoculant on the label. You can just whatever, you can just remember them as the first commercial mycorrhizae. So both of the 3 was purchased from the store, grocery store, or online, whatever. You can buy them like anywhere. And when I purchased them, they are solid. So they are just like powder or granular, but they are solid, not liquid, not air. So for the first one, bioorganics, I just added 10 milliliter of them. It's like 2 teaspoons. Here, I'll show you. Yeah, here is the depot, like the sandwich method. Here's the depot, and it was, yeah, you can see. It was added in this part. So I add like one third to one, like 50%, around 50%, but not more than 50% soil into the depot first. This part is bottom part, and then I add this. Use the teaspoon to add 2 teaspoons. Yeah, this is for first bioorganics. And also the second one.
Commercially, how are you applying it?
Oh, through this way, because I was applying the commercial mycorrhizae, use the same way as the description they gave me from the package. But you may apply more if you wanted to apply them in your field. Because
then you mix it with the fertilizer or you mix it with water. I'm trying to figure the approach of how to put it on rather than a test.
The first one and the second one, like the Indu and the bioorganics here, I just applied them. Oh no, I'm sorry. So the first one and third one, I just applied them. Like
Tianwen, I think maybe this might help. So the question is, if you were a grower, how would you apply these in the field? Would it be something you would apply to the soil? Would it be something you would put on your plants before you put into the ground? What would a grower do to apply these materials if they were going to put it on their farm?
If it's applied in the field, I think it's totally different. Yeah, in the like greenhouse. I think growers maybe mix them with soil, but I don't think
Mhm.
you can apply them like on the surface.
Right.
You should apply them like beneath the surface,
Yeah.
but you can like mix them with soil. And for the second one, in the description, it will be applied in the liquid. So in the greenhouse, I just dip my raspberry in the liquid for seconds, 5 seconds, and then transplant them in my deep pot. But if you apply them in the field, I am probably assuming you make like higher density of them to apply, because if you use too much water, they will leach from the soil.
So just to recap there, the first two are soil applied during planting, and then the second one, it's a root dip, also applied during planting.
I know it's really complicated, but if you feel interested about that, you can email me and I can like let you know like more details about them. Any questions?
All right. No, thank you. I think we're good.
Oh, okay. Thank you.